Good afternoon, or good evening, or good morning, everybody from all over the world. I'm from Japan, Sendai, and I'm happy to be here. First, I'd like to thank Chris and Lina and Giovanni for giving me such wonderful webinar SphingoLeader biology session. This picture is Mount Fuji, last December, when I flew from Sendai to Taipei to organize a joint glycobiology meeting, SphingoLeader meeting in Academia Sinica. Now, since then, I didn't have any flight, almost 1 year. I missed it. So today's talk is, my talk is homeostatic and the pathogenic role of GM3 ganglioside. Because my first career as a postdoc was development of the SphingoLeader inhibitor, so-called D3 or PDMP, which is a specific glucosylceramide synthase inhibitor in 1985. Then I back to Japan. Then I successfully cloned the GM3 ganglioside. This is GM3. So you may be better to recognize that this GM3 is a primary enzyme for the ganglioside biosynthesis, especially for the A and B3s. So if you block this enzyme, A and B3s is gone. On the other hand, 0,3's ganglioside is retained. Since we cloned GM3 synthase about 20 years ago, we generated a GM3 synthase deficient mouse. Then we learned a lot of phenotype if you give them stress. For instance, this is the insulin receptor. In the absence of GM3 synthase deficient, if you give the high-fat diet, they don't get insulin resistance. Also, today's main topic is amelioration of the chronic inflammation revealed by these papers. As you know, GM3 synthase deficiency is revealed in human. And before elucidating the hearing loss in human, we published the mouse as totally hearing loss, especially for the selective loss of the organ of Golgi, hair cells. Also, I will talk this one today, amelioration allergic asthma by selective suppression of helper T cell activation. I'll be back soon. And also, 2 years ago, this is normalization of appetite and leptin resistance in KKI mice. In the absence of GM3S, these mice did not get fat, keeping with their overeating appetite. Also, this knockout mouse background is ApoE-KO mice. These mice is normalized hypercholesterolemia in ApoE-KO mouse due to because NPC1L1, Francis Pratt is NPC1L12, but NPC1L1 is a cholesterol transporter located in enterocyte in a small intestine. So ganglioside, especially GM3, is needed for this function. So they cannot uptake for the cholesterol through this inhibitor. So I took two topics, this and this, due to the limited time. So I named after SphingoImmunology in innate immunity and adaptive immunity. First, I will give you for the CD4 and CD T cell, CD8 T cell require different membrane SphingoLeader for their development and activation revealed by this paper. So as you know, the TCR activation is believed to begin in the membrane microdomain, lipid love. Then after activation of the TCR, go to the immunological synapse, so-called central supramolecular activation cluster. If you cut vertical this activation cluster, this is a CS mark. PKC zeta or many other, CD3 or MHC molecule is concentrated in CS mark. Also, you can see the GM1 proved by the cholera toxin. This GM1 is a representative of the older ganglioside for the immunologist, but not for me. So GM1, what's the role of GM1? Proved by GM cholera toxin. I first interested in what's the difference or same in the individual T cell subset in the ganglioside species. So we take LC-MS global ganglioside species in the mouse thymocyte. This is a double positive 80%. Then peripheral primary CD4 and CD8 before activation. You can see the big difference with the L3's ganglioside. The thymocyte is really 8.4%, but CD4, 4%. CD8 cell, almost gone. On the other hand, 0-3's ganglioside, concentrating in a peripheral T cell subsets. Interestingly enough, this green is the thymocyte. This is RT-PCR data. During the developmental cascade from thymus to peripheral subset, CD4 T cell is GM3 synthase gene expression is double. On the other hand, CD8 T cell is downregulated. Then GM3 to GM2, GM2 GD2 synthase is almost 100-fold upregulated from thymocyte to peripheral T cell. So this is another LC-MS data. Thymocyte levels in individual GM1, GD1, Kiarnak GM1B, and extended GM1B. Extended GM1B also recognized by cholera toxin as well as GM1. But CD4 T cell, a little bit decrease of the A3's ganglioside as reviewed by this. But in the CD8 T cell, great enhancement of the 0-3's ganglioside. So I go back to why only CD8 T cell. Another keyword, sphingomyelin, is needed for CD8, CD4 T cells. So this is the first, elucidation of the mouse helper T cell and CD8 T cell, the distinct difference expression of the ganglioside species. So you can categorize CD8 T cell, CD4 T cell is A3's ganglioside dominant. CD8 T cells, O3's ganglioside dominant. So we take advantage of two knockout mouse, GM3 synthase knockout mouse and the GM2 GD2 synthase knockout mouse to review their individual ganglioside function in T cell subsets. This is GM2 GD8 knockout. Leave only GM3 and GD3. Other gangliosides should go on. But on the other hand, GM3 synthase knockout mouse is this one. So review A and B3 is deficient, but 0-3's ganglioside is maintained. Using these deficient knockout mouse, we took the CD3 and CD28 stimulated proliferation. In other words, TCR-dependent proliferation of CD4 T cell, CD8 T cell. So no stimulation, there are no proliferation, of course. But if you add anti-CD3 and the CD8 T cell, the co-stimulator signal, then you can see their wild type, great proliferation. But red, GM3 KO-derived CD4 T cell, nothing to proliferate. But GM2 GD2 synthase, which leave the GM3 and GD3, is normally proliferate. But this deficiency is completely recovered by the addition of A series ganglioside, GM1, but not 0 series ganglioside and GD1B. Opposite is true on the CD8 T cell. CD8 T cell is, you can see the green, GM2 GD2 synthase, which has only GM2 and GD3, also gone with the O series, 0 series ganglioside. Proliferation is completely attenuated. This inhibition was not rescued by A series ganglioside, but rescued with the 0 series ganglioside. So we did the series of supplementing study. Then we understand helper T cell is needed for the A series ganglioside, but not other, O series ganglioside. But CD8 T cell is required for the O series 0 series ganglioside, but not A series ganglioside. We so surprised such critical differences of the helper T cell and the killer T cell subset in terms of the requirement of the ganglioside species. This is a super antigen-induced, all activation, helper and the CD forever. This is also revealed. SAT1 is the GM3 synthase. This is selectively suppressed. CD8 T cell is, only selectively suppressed the GM2, GD2 synthase knockout mouse. The same are true for the mixed lymphocyte reaction. So we notice, nevertheless, the selective impairment of TCR signal, CD8 T cell and CD8 T cell in these knockout mouse-derived T cell, but intrinsic activity stimulated PMA and ionomycin was retained. So we next asked which signaling pathway is deficient in these knockout derived T cell subset. So you can see PMA ionomycin fully recovered, but anti-CD3, PMA ionomycin, PMA alone is completely recovered, but not ionomycin. Same are true in CD8 T cell. So we can conclude these knockout cell-derived helper T cell only deficient, not calcium signaling pathway, PKC signaling pathway. So how do we reconcile this kind of distinct difference? GM1A, A series ganglioside, and this is 0 series ganglioside rescued helper T cell, killer T cell. This only needed for the helper T cell. So discuss with Sandro Donino, University of Milano. GM1B is a straight structure using a very numbers of main conformers. It has 0 series redundancy. Highly moving capacity is bigger than GM1A. So this might be affect the microdomains organization. This could be the reason, but not yet proved. Take advantage of the proof for the pathophysiological significance of the GM3 synthase knockout mouse, because GM synthase knockout mouse helper T cell is selectively attenuated. So we take the experimental Azuma in GM3 KO mice, sensitizing the ovalbumin followed by the ovalbumin inflammation, then measured bronchoalveolar lavage fluid for cytokine, and also the cell type species in serum. So in the deficient, GM3 synthesis deficient, this is the path staining. There are no signs of the inflammation. Also, this is the individual macrophage and the eosinophile and neutrophil lymphocyte. So wild type, compared to wild type, GM3S is eosinophile infiltrating eosinophile is significantly reduced. At the same time, OVA-specific IgE is greatly reduced, with also reduced and IL-4 and IL-5. This is a crucial experiment with the pre-sensitized wild type to adoptive transfer experiment of the CD T cell to wild type, activated wild type. But if you transfer to the GM3 T cell, also proved for the eosinophile reducement. So this is a crucial proof for the T cell itself is functional deficient, this in vivo in the experimental Azuma model. So in the helper T cell, interactive the MC2 and CD4 and CD28, this TCR required acid ganglioside. We still don't critically understand what kind of specific specificity. Next is a developmental stage. So we conclude for the thymocyte, double positive to single positive, repertoire selection from DP, double positive to single positive cell in thymus, is accompanied with the selection of ganglioside expression in individual T cell subset. And this Greg-Spinglevit selection processes is indispensable for the distinct function of the mature T cell subset. So these findings may open up the strategy for targeting the helper T cell to treat allergic inflammation, including Azuma, by controlling the ganglioside expression in lipid raft. Also, this is unpublished data. So if you deplete all the ganglioside for the double knockout mouse, GM3S and GM2 DKO, we counted the cell number, DKO in thymus, is almost more than 100-fold reduced. Also, body is very small. Also, spleen and bone. So, initial defect found in the thymus. This number is comparable for the skid mouse, LC and fin knockout, and log KO. So, this means the gangliosides indispensable for the development of the T cell in thymus function, T cell development. So this is a precise picture of the development cascade from bone marrow in the thymus. So starting from the DM, double negative, double positive, and double positive to positive negative selection, called the repertoire selection. And then intermediate go to the single positive, and then periphery. So this is a technique for the flow cytomemory memory to segregate, distinguish all the T cell subset. So this is, expression profile of the cholerotoxin B, and recognizing GM1 and extending GM1B. CD8 T cell is required for the GM1 also is ganglioside, but compared to the CD40 cells. What about sphingomyelin? You can see, we surprised this TLC. Such a distinct difference for the DN and CD and DP and CDN in terms of the sphingomyelin levels. So this is 2 years ago, we found that DN and DP is very, very low. But CD4 T cell, as you mentioned before, the sphingomyelin level is greatly upregulated in the helper T cell, but not killer T cell. So this is, of course, the thymus is dominantly expressed SMS1, not SMS2. So in the SS1 knockout mouse, there are no expression of the sphingomyelin proved by the licensing. So this is, don't you interesting? You know, during the thymus development, ganglioside expression and sphingomyelin expression is very symmetrical. This is a very fascinating finding. So we, in this paper, due to the little short of time, the sphingomyelin is needed for the repertoire selection and positive and negative selection is on the way. But anyway, CD4 and CD8 T cell is a distinct requirement in terms of the ganglioside and the sphingomyelin. We should understand. Although it's a murine thymus, we really interested in human. So now we concluded the helper T cell is required A3s and sphingomyelin for the development and activation in the thymus. But killer T cell is required for 0 series ganglioside. We don't know the exact reason, but this is the truth in the murine T cell lineage. So, to summarize this picture, CD4 and CD8 T cell exhibit distinctive expression pattern of ganglioside 3 and sphingomyelin, which may lead to the effective immune function in each T cell subset. This is a current conclusion. We need to further experiment for the molecular mechanisms. So we're not going to sift the SphingoImmunology too, concentrating on the innate immunity in insulin resistance and inflammation in the metabolic syndrome. Almost 20 years ago, when I at the Tohoku University, I accidentally found leptin deficient OBOB, the leptin resistance, insulin resistance Zucker rat. When we analyze abdominal adipose tissue, GM3 level is greatly enhanced based on a GM3 synthesis messenger RNA expression. What's the reason for that? We did, this is 3T3L1 adipocyte. So to reveal what's the commitment of the ganglioside in insulin signaling in the state of insulin resistance induced by TNF-alpha. So when you treated the latent 100 picomolar TNF, very low amount, 96 hours. This is a world protocol to induce insulin resistance in this cell adipocyte. You can see insulin stimulation enhance the autophosphorylation, then followed by the IRS insulin receptor substrate tyrosine phosphorylation. But after treatment TNF-alpha, even after the insulin signaling, stimulation, IL-1 tyrosine phosphorylation completely attenuated. At the same time, as we can see the OBOB or Zuka, GM3 level is greatly upregulated, enhanced due to the GM3S synthase messenger RNA upregulation. Then we take advantage of the DPDMP, which I developed with Norman Redding and the University of Michigan. In the presence of DPDMP glucosylceramide synthase inhibitor, this increase is greatly attenuated, normalized, even in the presence of TNF treatment. But at the same time, insulin stimulation, insulin resistance is almost recovered in this condition. This is a great invention to applicate the glucosylceramide synthase inhibitor for the metabolic syndrome of some of the disease. This is 20 years ago, Norman Redding and me, we stayed and near the Stanford 3 days because he retired from Michigan to go to the Silicon Valley to work his son in Michigan, Silicon Valley. We enjoyed, but he now gone to the went heaven. So this PDMP is now, as Jim Shyman is developed and or Johannes Arts developed a strong inhibitor, and but this is I'm very happy. This PDMP is the start point for this field. So, take a long story short, in a normal adipocyte, the insulin receptor localized in caveolar microdomain. The insulin binding to insulin receptor, this stuff is concentrated on this area. So glucose transporter is for cytosol to group membrane, and glucose is level is reduced. But in the state of insulin resistance, insulin receptor is retrieved from the caveola to the other domain, which is localized called DIM, ganglioside enriched domain. So even insulin receptor to binding to insulin, autophosphorylation is induced. But after that, it's far from to following cascade. So this is a long time ago, more than 10 years ago, is a kind of famous picture for this intracellular insulin plasma membranes and insulin resistor cascade, because beta subunit of insulin receptor has a lysine residue. So GM3 is a carboxylic sialic acid. This strong ionic interaction is retrieved insulin receptor from caveola to GM. So this paper, also we analyzed the receptor tyrosine kinase family just above the sequence of the transmembrane region. Not only the insulin receptor, EGF receptor also possess the lysine amino acid just above the transmembrane region. So this paper interrogated for the Kai Simon's labs experiment. This is recombinant EGFR in a mammalian cell, which has a sugar chain also. This EGFR incorporated in a LD giant liposome or LD-LO giant liposome. Then see what happened. The LD disorder, only disordered, sphingomyelin cholesterol level is big, very low compared to the LD-LO phase. Interestingly enough, in the absence of GM3, in the absence of, without stimulation of EGF, EGF receptor already autophosphorylated in LD giant liposome. This is abnormal. But in the LDLO phase, is EGF receptor is activated by EGF in the dose-dependent manner. And then incorporated in the GM3. This is included in the ADP, the inside giant loop. And this one is the GM3 plus. So there are no effect of the LD, but the LDLO giant liposome GM3 is worked as a strong inhibitor of EGFR stimulation, only in the LDLO liposome. And finally, they also do like us for the insulin receptor. They switch for the K lysine to alanine, this region, in the normal inhibition of the GM by the GM3 in LDLO phase plus GM3. So this is, I hope, the receptor type tyrosine kinase, at least insulin receptor and the EGFR affect GM3 level in terms of the signal transduction. So we propose, this is a very famous intracellular insulin resistance mechanism proved by the Bruce Spielman at Harvard University. High-fat diet, especially for the paramedic saturated fatty acid, activate toll-like receptor. We're going to toll-like receptor soon. And activate GNK-IKK. Phosphorylated for the serine residue ILS1. This is the auto-inhibitor of the insulin signal. This is famous. In addition to this intracellular mechanism, we proposing increase the GM3 level by the inflammatory cytokine, at least adipocyte, dissociation of IL-caveolate complex. This is a double mechanism for the insulin resistance. So we proposing long time insulin receptor and insulin resistance as a membrane microdomain disorder. Not only the OB mouse, but also just high-fat diet for the B6 mouse and analyze the adipose tissue. We found a great increase of GM3 accompanied those high blood glucose levels. This is the reversible phase. So we gave the GM3 knockout background on the B6 mouse with high-fat diet, but happened to see. So only in a high-fat diet conditions, glucose tolerance test, the insulin tolerance test, is significantly, attenuated, normalized. If you give the high-fat diet, the insulin tolerance and the glucose tolerance is signaling is attenuated, but in the GM3 knockout, it is only in the high-fat diet conditions greatly attenuated, improved, ameliorated the state of insulin resistance and the diet-induced obesity. Body weight is normal, a little bit small, but GDT and ITT is improved. Also, we analyze the inflammatory cytokine and anti-inflammatory cytokines such as TNF and IL-10 respectively. So high-fat diet, they grow fat, obese, but in the absence of GM3 synthase, TNF-alpha level is decreased more than 50%. On the other hand, IL-10, anti-inflammatory IL-10 is increased, which means B6 mouse, the GM3 synthase deficient mouse get obese. However, wild type is adipose tissue is inflammatory condition by the activation of M1 macrophage. But this one as proved by the RT-PCR, IL-10, adiponectin, leptin. So these are anti-inflammatory environments still. So probably M2. So in this obese mouse of GM3 deficient mouse, get obese, but maintain the preservation of the adipose tissue function and insulin sensitivity. So keeping hyperplasia, but not hypertrophy. So HFD-induced obese GM3 KO mouse was ameliorated obesity-induced systemic insulin resistance and free from chronic inflammation in visceral adipose tissue. So why, before getting into the question, let me introduce our blood serum. It's more than 90% ganglioside is GM3. But GM3 is not only one species. Many isoforms, which are different for the N-acyl chain or hydroxyl chain, starting from the 16, 18, 20, so the long chain fatty acid, and the 20 to 24, and hydroxy 24, and the 24, and the double omega-9 double bond 24-1. Such a diverse structure is found at more than 30 species in our serum. This is the top 10, 16, 18, 20, long chain fatty acid. Very long chain fatty acid, 22, 23, and 24. Very long chain fatty acid plus double bond, omega-9, 22-1, 24-1. And hydroxy, very long chain fatty acid alpha hydroxy. What's meaning? Pathophysiological meaning. What's the difference of this kind of molecular species of GM3? This is just published EMBO. We take the patient with just BFA, visceral fat accumulation over BMI 30, but not sign of the metabolic syndrome. VFA plus hyperglycemia, VFA plus dyslipidemia, VFA plus both. These people with measured the molecular species of GM3. So interestingly enough, intramuscular thickness, a HOMA-AR sign of insulin resistance, BMI, positively correlate very long chain fatty acid. But on the other hand, HDL, good one, is positively correlated to 16 and 18. What's mean? So we plotted the long chain, very long chain, or unsaturated very long chain, is sharply decrease VFA with healthy, but VFA is a pre-symptomatic phase. But on the other hand, very long chain species gradually increase as the disease is progressing. So this is a heat map. Long chain fatty acid is decreased in VFA or lipidemia. But very long chain is gradually increased in the disease progression. So since GM3 synthetic knockout mouse is no more inflammatory in adipose tissue, so GM3 itself might be affect the innate immune system. This is our working hypothesis. So we check the innate immune receptor. We found, very interestingly, is the human monocyte primed, skewed through the M1 phenotype by the GM-CSF. Then put into the LPS primed level. This one, IL-6 is secreted. But in the presence of 5 micromolar physiological amount of very long chain, LPS priming is greatly accelerated. But on the other hand, long chain C16 and 18 and 21 is even in a priming effect of LPS is inhibited. Not only the LPS, HMGB1, it dumps, it's a very dangerous dumps for the activation of the innate immune signaling in the body. This is adipose tissue. Not, just lean and obesity, HMGB1 secretion from the adipose tissue is greatly enhanced in the human adipose tissue. So HMGB1 and SMR2, the very long chain, is synergistically activated for the LPS priming HMGB1 effect. But C1618 is suppressed, same as LPS case. The other, we look at the specificity, not only the only TLR4 is selectively affected by the GM3, but not the other TLR9 and TLR3 and 1 and 2. So this is a luciferase assay to see the activation of the NF-kappa B upon stimulation of the LPS. So we proved with NF-kappa B activation also synergistically activated the very long chain, but long chain is suppressed for the even in the LPS effect, and 24-1-2. Another thing, sorry, this is very important. LPS priming effect here. Then we added the C20 very long chain. So this is a synergistic effect. This synergistic effect with very long chain is cancer in the presence of long chain fatty acid, which means GM3 molecular species level is very keeper at the homeostatic or pathogenic condition in human beings. So this is TLR4 ligand, but this is pro-inflammatory. This is the anti-inflammatory. If you mix them, it's homeostatic. And this is a very busy picture, but this is the agonistic LPS. The antagonistic erythrin, which is now a clinical trial for the COVID-19. Also, lipid 4A, the antagonist. Then C16, to make a long story short, antagonist behave like a lipid 4A. And the very long chain is act as an agonist because, inserting the different orientation of the MD2 molecule. This is a simulation. 24 versus GM3 is the opposite way. Sphingosine and acyl chain is differently inserted for the MD2 hydrophobic pocket. Please refer to the MD journal. Also, LPS interacting basic amino acid residue chain to RNA. GM3 effect 22 is also reduced, suggesting the interacting LPS and the GM3 is similar way. Also, even in a nanomolar, 7 nanomolar or 1 micromolar, even this is a dimerization study for this page after cross-linker DMP. Very long, very small amount of the GM3 is induced dimerization, synergically induced LPS priming effect. So, imbalance of GM3 molecular species induced chronic inflammation through TLR4-MD2 complex activation. This is also cartoon for the embryo. So now, make a long story short, our normal condition, the pro and anti-inflammatory species is harmonized. But in obesity, pro-inflammatory, especially anti-inflammatory species decrease. This imbalance is activated chronic inflammation in the metabolic syndrome. So we propose a novel inflammation amplification loop triggered by GM3 molecular species and the new paradigm for the prevention of therapeutic intervention of metabolic syndrome, especially pre-symptomatic phase, because the decrease of the anti-inflammatory GM3 species, and even at this stage. So finally, GM3 ganglioside human serum is composed by a variety of fatty acids, including long chain and very long chain. And the serum VLCFA GM3 increased significantly, and LCFA GM3 decreased sharply in the metabolic syndrome at least pre-symptomatic phase. GM3 by itself had no effect on TLR4 activation. However, VLCFA GM3 synergistically and selectively augmented TLR4 activation by LPS or HMGB1, and in contrast, LCFA and unsaturated VLCFA GM3 suppressed TLR4 activation. GM3 interacted with extra regions of TLR-MD2 and modulated dimerization or mineralization. Ligand macromolecular docking studies suggested VLCFA and LCFA GM3 act as agonist and antagonist against the TLR4 activation respectively by differentially binding to hydrophobic pocket of MD2. So the VLCFA GM3 could be a risk factor for TLR4-mediated disease chronic inflammation. Thank you very much. And this is a collaborator in our lab, and this is a collaboration with University of Tokyo, Milano, University of Milano, Osaka University, and Gifu University. These are past member of our laboratory, friend member of our laboratory. They contributed tremendous in this research.